everyone, my name is Dr. Valerie Meyer-Spiridolosi, and I teach Industrial and Systems Engineering here at the University of Rhode Island. The Yukon URI Navy STEM Coalition works with the National Institute for Undersea Vehicle Technology and our partners throughout the region who build submarines and support the U.S. defense industries. Come take a look what we're doing.
Hello, everyone. My name is Dan Roxbury. I'm Associate Professor of Chemical Engineering. I'm also the Graduate Director in the department here at the University of Rhode Island. Welcome to my lab. This is our URI NanoBio Engineering Laboratory. The focus of our lab is developing new types of imaging agents and sensors based on novel nanotechnologies. My graduate student, Asir Nadim, he's going to give you an overview of nanoparticle preparation, including preparing carbon nanotubes for biomedical applications. And my student, Ella, she's a junior undergraduate chemical engineering student, and she's going to give you an overview of our smart bandage applications.
Hey everyone, my name is Asir. So what we do in this lab is a lot of interesting science with nanotubes. The question is, what is a nanotube, right? This is a model of a nanotube. So basically, a nanotube is a hollow cylinder made up of single layer carbon. What do you think will be the actual size of these nanotubes? You can make these nanotubes according to the size you want. So anywhere from 1 nanometer to 100 nanometers, which is actually really exciting. So we use these nanotubes to make different types of sensors. Sensors for cancer, sensors for wound healing, and a lot of different diseases, like a lot of brain diseases as well. We can also use these nanotubes for drug delivery. Now the question arises, these are actually synthetic. So how will these be able to interact with living things? So we wrap these nanotubes with DNA. The reason why we wrap these nanotubes with DNA is to make them more biocompatible. So the cells that we use in our experiments are happy, so they don't die and they keep growing. So we use this device to make our nanotube samples that go into the cells. Now this is called a sonicator. We start off by suspending these nanotubes in low concentration salt solution and add DNA to it. After that, we add those nanotubes into this device. So basically what happens in this device is you have a single strand of DNA and then you have a nanotube. With this device, we can wrap the DNA on the surface of a nanotube, like this. The advantages of wrapping them with DNA are that the cells can easily eat them up. With other synthetic polymers, they might go in the cell, they might not. The cell might eat them, the cell might not like that polymer. It's just going to stick to the surface and not do anything, and eventually kill the cell. After our samples are ready, we can add them to the cell cultures and do experiments.
Hi, I'm Ella. So here at the NanoBio Engineering Lab, I work on creating a smart bandage. Our goal here is to take the typical Band-Aid that we have and to make it be able to detect the progress of a wound's healing. When you get a wound, what do you do to help it heal? Your body releases this drug, hydrogen peroxide, which is actually naturally found within your body to help the wound healing process. For the applications of our bandages, they can be used as a sensor for the detection of the hydrogen peroxide. How do you think the carbon nanotubes are being utilized within this bandage? So what's special about our bandage right here is that it can detect how far along our wound has healed. So to do this, we have our single-walled carbon nanotubes within this very thin bandage. And these nanotubes are used to be able to detect how much hydrogen peroxide is within that wound. The reason why we're detecting hydrogen peroxide is because it's a natural component found within your body. Hydrogen peroxide is a sign that your body needs healing. It's a marker within the wound that shows when there's inflammation. There's going to be more hydrogen peroxide at a wound. Now, as the wound heals, the hydrogen peroxide concentration is going to decrease. So current bandages, you have to take them off to see how the wound is healing. The goal of our bandage is to be able to detect this hydrogen peroxide for up to 21 days, which means you don't have to remove it from the wound. What do you think the benefits of not having to remove a bandage would be? So So this big device behind me is what we use to make our bandages. So this machine uses 2 liquid solutions that, when a voltage is applied, becomes a solid like this. So they flow out onto the spinning drum that you see here and become a solid and form this bandage. So along with our bandage being able to detect the healing rate of this wound, we're also looking to be able to deliver healing drugs to the wound. If you had access to this technology, how would you use it to benefit your own community?
So thank you guys for taking a tour through my laboratory. If you're interested in improving modern medicine with new types of nanotechnology, I encourage you to think about URI for your undergraduate studies. Thank you.